Hello everyone, I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Characterization of CRISPR Technologies using TXTL and the Echo Liquid Handler, and our sponsor is Beckman Coulter Life Sciences. For today are Dr. Vincent Noireaux, a professor at the University of Minnesota, and Aset Khakimzhan, a PhD student at the University of Minnesota. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Noireaux. Please go ahead.
Thank you, Christy, for this introduction. My name is Vincent Noireaux. I am a professor at the University of Minnesota, and I will present this webinar with Aset Khakimzhan, a PhD student in my laboratory. This webinar will cover the four following parts. First, I will introduce cell-free gene expression, a relatively new technology to do some synthetic biology and bioengineering in vitro. I will then introduce and present the Echo Liquid Handler from Beckman Coulter Life Sciences. And then Aset will cover the third part on characterizing CRISPR with TXTL and the Echo Liquid Handler. And we will conclude this webinar with some suggested literature. Let me start by some terminology. There are many different ways to define the in vitro synthesis of proteins from DNA. CFE for cell-free gene expression, TXTL for cell-free transcription translation, IVTT for in vitro transcription translation, CFPS for cell-free protein synthesis. As you will see in this webinar, cell-free transcription translation has become a popular laboratory research tool in biology, bioengineering, synthetic biology, and biological physics. Major applications of TXTL include biomanufacturing proteins and prototyping DNAs from regulatory elements to gene circuits, as you are going to see with CRISPR in this webinar. Combined with automated liquid dispensing, TXTL can dramatically accelerate synthetic biology applications. Beckman, the sponsor of this webinar, provides a unique automated acoustic liquid handler ideal for TXTL work. In this webinar, we will use two terminologies, CFE and TXTL, to define in vitro synthesis of proteins from DNA. Let me introduce what is cell-free gene expression in this first part of the webinar. A cell-free gene expression system includes different parts. One of the major parts of a cell-free gene expression system is a cytoplasmic lysate prepared from living cells. Typically, we use E. coli to prepare a cell-free gene expression system, but other types of cells can be also used. The E. coli strain, the E. coli cells are grown in relatively large flasks to produce the cells, from which a lysate is prepared by breaking the cells and extracting the cytoplasm that contains the molecular component for transcription and translation. Two other buffers are necessary to perform cell-free gene expression reactions. An energy buffer that contains the ribonucleosides, such as ATP and GTP, for the chemical energy, and an amino acid mixtures that contain the 20 canonical amino acids. When mixed together, you can add your DNA, your plasmid or linear DNA, to produce proteins. Cell-free gene expression reaction can be performed at different scales. It can be performed in relatively large flasks, from the milliliter to the liter scale. It can be performed at the microliter scale on well plates or test tube. And as you are going to see in this webinar, relatively quickly using liquid dispensing from the microliter to a few tenths of microliter scale. And it can be performed from the femtoliter to the microliter scale in some microfluidic chips and synthetic cell systems. In this slide, you have an overview of the TXTL application and its scope. You choose a vessel like you see on the top left, either a test tube or a well plate. You prepare your DNA, you clone your DNA, your gene of interest. You prepare your plasmid or your linear template. You have your TXTL mix, and when you mix both, you produce proteins over incubation times of a few hours, up to 20 hours, at incubation temperature that goes typically from 29 Celsius to 37 Celsius. And after incubation, you get your product or your application. There are three main applications in cell-free gene expression synthetic biology. The first one is prototyping and biomanufacturing. With a cell-free gene expression, you can rapidly test and prototype DNA parts and gene circuits, enzymatic pathways that contain different enzymes. You can also biomanufacture biologics like antibodies and pharmaceuticals. And finally, you can also perform unnatural chemistries like introducing non-natural amino acids into your peptide or your protein. The second major application of cell-free gene expression is self-assembly nanotechnology. You can express and make virus-like particles. You can make phage parts and all phages. You can direct some self-assembly, and you can also study some new self-assemblies, as you can see just below in this electron microscopy image, where the phage T7 was entirely expressed and synthesized. And finally, the third main application of cell-free gene expression is synthetic cell and material sciences. You can use a cell-free gene expression system. You can encapsulate it in very small volume, as you can see in the fluorescence microscopy image at the bottom right. You see two liposomes of about 10 to 20 micrometers in diameter, where a cell-free gene expression system was encapsulated and used to express the MREB cytoskeletal protein from E. coli that polymerizes at the inner membrane. With such a system, you can study cell mechanics. You can build artificial cells by incorporating non-natural molecules. You can develop diagnostics. You can create some microchip. And more broadly, you can develop biotic-abiotic interfaces. Cell-free gene expression combined with liquid handling, like the Echo from Beckman Coulter Life Sciences, is very useful for two of these applications. For prototyping and biomanufacturing, as you are going to see with CRISPR in this webinar, and also for self-assembly and nanotechnologies. In this webinar, we will present some results that have been obtained using the cell-free gene expression kit called MyTXTL from Arbor Biosciences. It is a highly versatile and powerful cell-free gene expression system. It is simple to handle. You just add your DNA to the mix to produce your protein. It has a high performance, about 2 to 3 milligrams per ml of protein produced. You can produce protein with sizes ranging from 10 kilodalton to 200 kilodalton. It has a great versatility as it is compatible with many different promoters. It has a quick turnaround, takes just a few hours and sometimes just a few minutes. And finally, it has an all-time flexibility, as you can use both circular and linear DNA. In the second part of the webinar, I will present the Echo Liquid Handler from Beckman Coulter Life Sciences. There are two different models, the Echo 525 and the Echo 650. The volume of the droplets that are dispensed is 25 nanoliters for the Echo 525 and 2.5 nanoliters for the Echo 650. The speed of dispensing for the Echo 525 is up to 6 microliters per second and up to 1 microliter per second for the Echo 650. The Echo 525 is ideal for reaction of about 10 microliters. It can be smaller and it can be larger. And the Echo 650 is ideal for reaction of volume of about the microliter scale. It can be sub-microliter, and it can be a few microliters. The source plate used for the Echo 525 is a plate of 384 well, and you can dispense from this source plate into destination plates that are 6 well and above. With the Echo 650, the source plate can either be a 384 well plate or a 1536 well plate, and you can dispense in a wide variety of well plates from 6 wells up to well plates of 1536 wells. As you can see on this slide, the technology is based on acoustic. It is tipless, and there is no contact with the liquids. In the top left corner, you see the source plate with 2 different solutions. One that has a color purple, and the other one is yellow. Just below the purple solution, you see the acoustic system that is going to produce the small droplets that are dispensed into the destination plate. On the right, top right here, you see the source plate with a different solution of purple and yellow wells. And you see the destination plate upside down just above the source plate where the droplets are dispensed. And at the bottom of this slide, you see a time lapse of the formation of the droplets that are dispensed into the destination plate. Depending on whether you use an Echo 650 or 525, the speed of dispensing can go up from 6 microliter per second for the 525 and 1 microliter per second for the 650, which represents about a few hundreds of droplets per second. How the Echo liquid handling really works, here are some additional characteristics and schematic. It is an acoustic energy. The volume of the droplet is 2.5 nanoliter for the 650 and 25 nanoliter for the 525. It has a high accuracy and precision. It has a low energy transfer, and it preserves the sample integrity. As you can see on the right here, you have a source plate at the bottom with 4 different solutions labeled as A, B, C, D, blue, green, purple, and red. And just above, upside down, is your destination plate where the liquid is dispensed. What you have to understand is that you can create whatever combination of the solution on your source plate into any of the destination well. You can mix liquid A with liquid B, with liquid C, with liquid D at whatever volume. The dynamic of the fluid analysis, you can adjust the transfer parameter in real time for a variety of fluids. You can use various samples and reagents at different concentrations without any user calibration. And very important, the Echo liquid handler is compatible with a very broad and wide range of liquids, and in particular biological fluids, DNA solution, RNA solution, enzymes, master mix in glycerol stock, and you will see in this webinar it is compatible with cell-free gene expression solutions that are very rich in polymers, in protein, and in small molecules. A TXTL reaction can be prepared either by hand on the left in this slide or using the Echo liquid handler on the right. You typically have a TXTL mix. You have typically some nutrients to add. You have your DNA solutions ready, and you have your water. When you use pipettes, you typically obtain a relatively large error in your reactions. It means that you will see some reproducibility in your work, but they will have relatively large error from one reaction to the other one. So the reproducibility is kind of limited when you use mechanical pipette like shown on the left here. Of course, you cannot do high throughput work when you use manual pipettes. There is a human physical limit. You are not going to do hundreds of thousands of reactions by hand in a day. or even in a week. And it takes, at the end, a lot of time. And finally, because of the pipette minimum volume, your cell-free reaction have a typical volume of 10 to 12 microliters. On the right, using well plates and the Echo Liquid Handler, your error will be small. It will be highly reproducible. Of course, you will do some high throughput work. You will be able to do hundreds of reactions in a single day. There is no physical limit. It consists of programming the machine. It's about 5 to 10 times faster than manual, and you are saving some material because your reaction volume can be at the scale of the microliter. With it conclude the second part, and I will let Aset present the third part and give you some example of using cell-free gene expression and the Echo to characterize CRISPR technologies.
Thank you, Vincent. So CRISPR technologies. CRISPR just absolutely revolutionized molecular biology in the last decade. And the beauty of CRISPR is that it can be used to target at this point, nearly any site on the genome or a piece of DNA. So initially that was thought that it could be used to do genome editing, and there are now many trials, clinical trials, to see how CRISPR can be used to edit genomes. But in addition to genome editing, it can be used to control transcription by simply mutating the Cas enzyme, so in this case Cas9. We can make the Cas9 enzyme incapable to cleave the target DNA, and now it just targets, and so it would bind to somewhere on the genome or on the gene, and that would either inhibit or activate the transcription. It can also be used to perform epigenetics, and by targeting Cas9 and combining it with some epigenetic protein that would regulate the DNA, the structure of the DNA, we can modify the epigenome of the epigenome. CRISPR-Cas9 can be used to perform genome-wide screens, and finally CRISPR-Cas9 can be used to do imaging. So the way we use CRISPR is mostly controlling transcription, and so here's I just wanted to show the workflow of TXTL with an Echo Liquid Handler and how we would get a CRISPR reaction with TXTL. So first we have to get the parts. So the DNA would be encoding for the CRISPR enzyme and the guide RNA and the target. We would load the lysate, so the TXTL, the resources, this DNA that encodes for all the parts, and some water into the destination plate. We would have our source plate ready. Afterwards, we load the destination plate into the Echo Liquid Handler, and from the destination plate, all the liquids would be spread into the source plate, and we would have our TXTL reaction done. So the dispensing takes 20 minutes, usually takes less. If we're doing not a full plate, it can take much less, 10 minutes or so. The next step would be to take the source plate and put it into a fluorescence measurement machine, and we would measure the fluorescence of a reaction. Because the targets are normally some kind of fluorescent protein, we can see the activity of a CRISPR-Cas9 enzyme or some other CRISPR enzyme based on how much DGFP, or which green fluorescent protein, or some other fluorescent protein is being expressed. And so by the level of expression, we can tell how well CRISPR works. And last, so that takes about 4 to 12 hours. And last, we get our data, we have our fluorescence measurements, and we can analyze it, and we can get our answers. So that takes 1 to 2 hours, but if it's a repeat experiment, it can take almost immediately. So just to show, recap how we do TXTL CRISPR. So here's an example of CRISPR-Cas9 and TXTL. We have a plasmid, so a plasmid that would express Cas9 or Cas9, Then we have a linear piece of DNA that would express the guide RNA. Here it's shown as sgRNA. And then we would have our target. So in this case, it's DGFB. So we have these free proteins, free genes that express their proteins or RNA. And then we have our TXTL components. So that would be the lysate, the amino acids, the energy mix, and water. We take all of this, we put it into the source plate, it gets dispensed. We have a destination plate that is ready for measurement. And in this case, we have what we did in the plot below is we changed the guide RNA. So in one of the examples, the guide RNA is targeting the promoter site, so that's SG6. Then there's another guide RNA that targets SG3. That's also a promoter, but closer to the gene. And then the last guide RNA targets the gene itself. And you can see that by changing the guide RNA, we can notice some differences in the silencing of CRISPR. So the green line would be the control. So that's just the gene not being silenced. We would be using some non-targeting guide RNA. And then the blue lines, the blue plots, are the expression when the gene is being targeted. So we would be interested in looking at where on the promoter plus gene should we target. So what kind of sequence should we take for our guide RNA, and what should we use to silence the gene in the most efficient way? And we're not only interested for that in TXTL, we would also like to see, does the silencing in TXTL agree with silencing effects in vivo? So now we take a lot of different guide RNAs with different qualities to them, different PAM sites, or are they template or non-template targeting? And because there's a lot of these different guide RNAs, it would take a long time to check all of them, doing the reactions by hand. But as Vincent specified before, doing these reactions with an echo would be 5 to 10 times faster, depending on if it's a repeat experiment or a new experiment. And so we use the echo, and we can measure the efficiency of all the guide RNAs very quickly with it. And as you can see in the plot below, we have our y-axis for the repression in E. coli, and we have our x-axis for the repression in TXTL. And there's an excellent agreement between them. And all of that, so the x-axis experiments are done very quickly. This is probably 3 to 4 plates. That's a couple of hours of a workday. And the y-axis, so that's done in E. coli, that's multiple days of work there. And so we can measure very quickly the efficiency of a guide RNA using TXTL, both in vivo and in TXTL. We can also do that with other CRISPR enzymes. So there's the Cas3, the Cas3 enzyme, the cascade enzyme, cascade set of enzymes which form this cascade complex. Or we can look at Cas12, which is also a single protein CRISPR system. And we can do the same thing and see their efficiency in TXTL. So here's an example of Cas12. We express the Cas12 protein, we express the guide RNA that corresponds to the Cas12, and we express DGFB. And here you can see how Cas12 works. We can do the same thing for Cas3. Cas3 also targets DNA, and so here we express the proteins, the cascade, and we can look at how that works. And here again, green line is the control, blue line is the measurement, and we can see how Cas3 works. So in addition to Cas9, TXTL can be used to measure the efficiency of other CRISPR proteins. Recently, we've also been looking at Cas13, and then there's a whole slew of different Cas enzymes and their variants for mutants, Cas enzymes from other organisms. So TXTL is excellent for that, and it can be used to prototype CRISPR enzymes and their guide RNAs. In addition to CRISPR proteins, there's also the anti-CRISPR proteins. These are the proteins that would inhibit the effect of CRISPR enzymes. So as you can see in this example, ACR2 is an anti-CRISPR protein. It can take the CRISPR-Cas9 complex and bind to it and when it binds to it, it prevents CRISPR-Cas9 from binding to the gene. And that way, the anti-CRISPR protein would inhibit the effect of CRISPR. So what we would do in TXTL is, in addition to expressing Cas9, the guide RNA, sgRNA, and DGFP, we would also express the purple linear, here it's purple, this linear gene, which encodes for ACRs, the anti-CRISPRs. Anti-CRISPRs are normally very small proteins, and so we can put them onto linear DNA very easily and just order them online and get a lot of different anti-CRISPRs to test. And so we have our reaction, we put all of this into the source plate with the TXTL components, and we get our measurements. And so in this case, in the same paper done by Ryan, Noireaux, and everyone else, you can see there's 24 different anti-CRISPR proteins, and there's 5 different Cas9 enzymes. And so for each Cas9 enzyme, we checked which of the anti-CRISPR proteins would work. And so that's 24 times 5, and for each of them, you have to do replicates. So 24 times 5, that's 120 different reactions. And so if that had to be done by hand, that would take a lot of time. But instead, this is all done with a Echo Liquid Handler. And because of that, the whole process gets way faster. And you can see there's stronger inhibition or weaker inhibition depending on which ACR is coupled to which Cas9 enzyme. So in addition to changing the location where the guide RNA targets, we can change the guide RNA itself. So we can introduce truncations to the spacer sequence of a guide RNA, or we can introduce mismatches to the spacer and see how that would work. And so here we have the characterization of Cas9 guide RNAs using TXTL. And so here you can see there's this reaction. We express dCas9, we express a guide RNA, we express GFP, but in this case, the guide RNA has these different changes. So we have a spacer sequence that is from 5 to 32 base pairs long. And then we have our TXTL reaction, and again, we start off with a source plate, we load everything into the Echo, and we get our measurements. And so these are the measurements, and as you can see, we have these, the x-axis is the length of the spacer sequence on the guide RNA. The y-axis is how well it targets, how well it silences. So the higher the number, the better the silencing. So it binds really well. And we measured the strength of silencing for different spacer lengths, for different concentrations. And we noticed something very, very weird, that the shorter spacers performed on the same level and sometimes even better than the conventional 20 base pair spacer sequence. And to just confirm these results and get a very good measurements, we did even just to get a good view, are we wrong? Is this actually the case? We performed a lot of different measurements. And here in D on the bottom right, we got the binding curves for all the different guide RNA lengths and checked if we were correct. And so here we have the plot C, top left. This is KD for all the guide RNAs. And indeed, in fact, the guide RNA, the KD is much lower for the 10 base pair spacer length than it is for 20 base pair spacer length, which is very surprising. But the big part here is that we had to do a lot of measurements to get this plot. So for this plot alone, we probably had to do 1,000 TXTL reactions. Now with a liquid handler, I remember this took me, I did it within a working day, so about 6 hours. And if I had to do all these experiments by hand, it would take me probably about a week of just working nonstop, going in and doing reactions every day. And just a recap of how much faster doing reactions with an Echo is than doing it by hand. So we have the plots on the bottom. So on the bottom left, if we are doing a new reaction, that means we have to program the reaction in the Echo. Then it's about 5 times faster to do it with an Echo. But if a reaction is a repeated one, that means we don't have to program it. It's already on the computer, and we just have to launch the dispensing. Then the reaction is 10 times faster. And in this case, we had to do a lot of repeated experiments. So having an echo helped a lot. So it made it way faster than it would have been by hand. And finally, this is something that we started doing recently, is using CRISPR-Cas9 or other CRISPR enzymes to construct genetic circuits. So because CRISPR-Cas9 can be used to silence genes, that can be used to, so that's basically repression. Or we can use it to activate genes also. And so in this case, we have an IFFL, an incoherent feed-forward loop, and this is an unpublished paper written by Ben Tickman, and it's going to come out soon. And here we studied how we can use CRISPR-Cas9 to make circuits. And the initial stages of building these IFFLs, we had to do a lot of experiments with the echo. And so we have these 4 genes. You can see X, Y, Z, and Y dash. X, Y, and Z, and Y dash would be a separate experiment, which is the control. And so by changing all the concentrations of all these genes, we have a lot of complexity. So how much of X do we need? How much of Y do we need? How much of Z do we need? So that's a lot of experiments, and we have to look at the whole parameter space. And if we do these reactions by hand, looking at the parameter space takes a while, but doing it by echo, it's faster, and so we can get the specifications of a circuit quicker. And with that, I'm done with part 3, and I'll give it back to Vincent.
Thank you, Aset, for showing us how to combine the Echo Liquid Handler, cell-free gene expression to do some rapid characterization and prototyping of CRISPR technologies. In this slide, we suggest some literature, some articles with the 3 components, CRISPR, cell-free gene expression, and the Echo Liquid Handler, and some references about cell-free gene expression in general. You have 2 reviews here, 2 recent reviews, covering the applications of cell-free gene expression and an introduction to cell-free gene expression. Results that we have shown in this work have been obtained by students and postdocs in the laboratory of Vincent Noireaux at the University of Minnesota. If you want to know more or you have questions beyond the webinar, we have the contact information on this slide, and the results that we have shown in this webinar were sponsored by the following agencies.
Thank you, Vincent and Aset. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. And joining us for the Q&A today is Sohaila Beck, Commercial Product Manager for Genomics at Beckman Coulter Life Sciences. All right, let's start the Q&A. Okay, so Vincent and Aset, our first question, are the ultrasounds used in the Echo Liquid Handler strong enough to shear DNA?
So that's a very good question. I'm going to have Aset response to this question because he is using the Echo almost every day, and he knows very well how to respond to this question.
No, it doesn't shear the DNA. Never happened. If the energy is just not enough to cause any breaks in the DNA.
Great.
I would add something with respect to what just Aset said. That for TXTL, mostly so far we have used plasmid DNA of several kilobase pair. A typical plasmid is 4 to 5 kilobase pair, and we have also used linear DNA of a few kilobase pair in size, and we have never observed any major degradation or shear of the DNA when we dispense it with the Echo Liquid Handler from Beckman Coulter Life Sciences. That's it.
Great, thank you, Vincent. So then our next question is, have you noticed a concern at all for DNA or RNase contamination with TXTL reactions? Because it seems that that could significantly affect CRISPR readouts.
That's a very good point, because when you dispense with the Echo, the two plates are in the Echo. They face each other. And in particular, the source solutions that are on the source plate, everything is open, of course. And we have done hundreds of plates dispensing with the Echo, and we have never seen here also major issues of contaminations or things like this. Now, on a regular basis, we have a outlier in the reactions. Typically on the plate we do per condition 3 or 4 repeats, and sometimes one of the repeats out of the 4 repeats shows a completely different signal, but that is absolutely normal. It's not necessarily, that does not necessarily come from the Echo. It's probably when we close the plate after it is returned by the Echo, we have to put a lid. Or originally in the destination plate there was some dust or things like this. So there are not more outliers than when you do the reactions by hand. So the Echo does not introduce any major issues at that level.
And could you tell us what you found to be any practical limitations or any sort of less likable features of the acoustic liquid handler? Anything you might improve? For example, does it generate aerosols?
Say that again. Does it generate what?
Aerosols.
Aerosols. Not really. No. We have not. Really observed this. Maybe Aset can also answer this question. There are always things that you can improve. The Echo does so much already, and it's far better than what you can do by hand. What you have to take into consideration, for example, is that there is a dead volume. So systematically, you are going to load your solution on the source plate, and the Echo cannot use entirely the solution. There will be some leftover, but so far from us, it's not really a big deal. It's not a problem. It's relatively small compared to the volumes that we have and that we use. That's it.
Yeah, I agree. For the job, not a lot of complaints. As Vincent said, there's always something you can improve, but for a liquid handler, it does liquid handling very well, and so not a lot of complaints. Nothing substantial.
Great. Sohaila, did you want to add anything?
Hi, yeah. I wanted to say the aerosol question comes up quite often among customers, and we have run qPCR experiments, and there are papers on qPCR being done by Echo, by Big Pharma. I can point out, if you want to be contacted after the webinar, to those papers to show the quality of data for those qPCR sample preparations were actually better than traditional methods. So happy to get in touch and send you those literature to show the concerns about how acoustic ejects the drop and could probably cause aerosolization is not really a valid concern because the data shows otherwise. But great question. Thank you for the question.
Great. Thank you, Sohaila. So Sohaila, another question for you. How often should a user calibrate the Echo Liquid Handler?
That's another good question. In fact, Echo is the simplest liquid handler I've used myself. It doesn't need any calibration. We do a focus check every week to make sure the system is still in good health, and it pretty much always is. If an instrument is getting old and anything concerns someone, that focus check every week will tell us if anything needs to be checked. Otherwise, that's just basically like a quality check just to make sure everything is in shape. Other than that, a user chooses a liquid calibration, and it's not like you have to change the speed or you need to change any parameter. All of that work is done in manufacturing and by our engineers to find the right liquid calibration for a wide liquid class. I think Vincent mentioned that they have no problem transferring all these solutions for the cell-free extracts. Therefore, there is no calibration needed. It's not like a tip-based that you want to go and adjust the height of the tip or anything. You put your source plate in, you put your destination plate in, you choose your liquid class, and shoot the drop.
Great, thank you. Vincent, is it possible to dispense living cells using the Echo?
Yes, but typically the Echo is used to dispense some solutions, some liquid solutions. It turns out that you can also dispense living cells. We have done this a little bit. I know that some other groups have done this also a little bit. And the living cells, they stay living cells even after dispensing. There is no problem about this. That does not kill the cells. And you can dispense the cell like you dispense a liquid solution. So there is no problem at that level.
And are there any big differences in the cell-free expression of genes from linear templates and plasmids?
Yeah, that's also a very important question because using linear DNA has a lot of advantages because you can use PCR products, for example, and it is absolutely possible. Gene expression from cell-free gene expression from linear template is not as strong as from plasmid. I would say on average it's about 60 to 75% of the cell-free gene expression that you get from a plasmid. So it's still very good and very practical. We do that on a regular basis in many of our reactions. We have plasmids, we have linear DNA. And that works very well. What you have to know is that linear DNA in a typical cell-free gene expression system is degraded very quickly. And there are methods that have been published, very easy to implement, to use, that protect the linear DNA, and therefore you can do cell-free gene expression and produce proteins.
Great, thank you, Vincent. I show no other questions in the queue, so I'm going to wrap it up there for right now. We'd like to thank Vincent Noireaux, Aset, Khakimzhan, Sohaila Beck, and our sponsor, Beckman Coulter Life Sciences. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. And as always, if you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.